In this class, let's look at a few typical reactor configurations for biological nutrient removal and their performances. The modified Luzak-Ettinger process is the most simple configuration for nitrification and denitrification. It starts with an anoxic zone, followed by an aerobic zone. The mixed liquor from the aerobic zone recycles back to the beginning of the anoxic zone. The anoxic zone is at the beginning because denitrification requires organic carbon as electron donor. If it is the other way, the organic carbon would be oxidized first, leaving no electron donor for denitrification. The electron acceptor for anoxic zone, nitrate, is provided by recirculating mixed liquor from the aerobic zone. This table shows the advantages and disadvantages of the MLE process. Because the nitrate removal in this process relies on the recirculation of mixed liquor, certain amount of nitrate always ends up in the effluent without treatment. It is not possible to achieve thorough nitrogen removal in MLE systems. There's always between 5 and 10 mg per liter of total nitrogen in the effluent. Here we can compare the effect of SRT on the performance of MLE system, represented by the solid line, with a single CSTR with the same volume and flow rate, represented by the dashed line. You can see the nitrate removal by the MLE system in panel C. As for the removal of soluble organics and ammonia in panels A and B, MLE generally needs a longer SRT compared to the single CSTR. This is because the aerobic zone for nitrification and heterotrophic growth is only half of the reaction volume, therefore shares half of the SRT. Also, some soluble organic carbon is removed through denitrification, where nitrate is used as electron acceptor. This does not generate as much free energy as when oxygen is used as electron acceptor, resulting in slower growth. Again, a lower oxygen requirement is observed in the MLE system because nitrate is used as the electron acceptor in the anoxic zone. This figure shows the effect of recirculation ratio on the performance of the anoxic zone, represented by the solid line, and the aerobic zone, represented by the dashed line. The higher the recirculation ratio, more nitrate produced from the aerobic zone is denitrified, resulting in a lower nitrate concentration in the effluent. But this also means more intense energy requirement for the recirculation pump. As for the oxygen demand, more recirculation results in more denitrification, therefore less oxygen is needed. What about the effect of volume ratio between the anoxic zone and the aerobic zone? The volume ratio is also the SRT ratio of these two zones. If anoxic zone is too large, there's not enough SRT for nitrification, and eventually not even enough for aerobic degradation of organic carbon. But if the anoxic zone is too small, there's not enough denitrification, and the effluent nitrate concentration will be high. To maintain an ideal overall nitrogen removal rate, the aerobic zone typically is 50 to 70% of the overall volume. This is also reflected on the oxygen requirement and nitrate uptake. An unbalanced reactor volume ratio can lead to drastic decline of nitrate utilization rate. The four-stage Barton flow aims to deal with MLE system's intrinsic issue of high Tn concentration in the effluent by adding another anoxic aerobic cycle after the first one, so that unreacted nitrate from the first aerobic reactor enters the second anoxic zone for further denitrification. The soluble substrates produced from the slow hydrolysis of particulates are also used as electron donor in this zone. And the final aerobic zone scrubs the residual soluble substrates and ensures high settleability. The four-stage bottom flow system can achieve very low Tn concentration in the effluent, but at the expense of large reactor volume. Again, let's look at the effect of SRT on the performance of the four-stage bottom flow system and compare it with MLE system, which is represented by the dashed line. Their removal of soluble organics and ammonia and production of biomass are very similar as shown in panels A, B, D, E, F. But because of the additional anoxic aerobic cycle, the four-stage bottom flow achieves a lower nitrate concentration in effluent compared to the MLE system. The oxygen requirement and the solid wastage rate of the four-stage bottom flow system is also very similar to the MLE system. The four-stage bottom flow system shows a slightly lower oxygen requirement, possibly because the further denitrification. We have also covered the concept of sequential batch reactor. It is like PFR over time. The reactor is first filled with influent to mix with previously settled biomass. The aeration or mixing can be controlled as needed in the reaction phase to keep the reactor aerobic, anoxic, or anaerobic. Once reaction is complete, the biomass is settled and the supernatant is removed. The reactor is ready for the next batch. Typically, several SBRs are operating at different stages in parallel to receive a continuous influent. SBR can be quite flexible. To achieve nitrogen removal, it can mimic the MLE system with one anoxic aerobic cycle or a four-stage bottom flow system with two cycles for a high effluent quality. To mimic the mixed liquor recirculation, a portion of nitrate-rich supernatant after the aerobic reaction is left in the reactor to mix with the influent with abundant organics for the next batch. This is equivalent as recirculation of mixed liquor. This graph shows the concentration of ammonia, nitrate, and COD in the SBR mimicking the MLE system. COD starts to be consumed for denitrification during the fill phase. The increase in soluble organics concentration in the aerobic phase is due to faster hydrolysis of slowly biodegradable or particulate COD compared to anoxic hydrolysis. Can you pause this video and think, how can this system be optimized? Okay, because there is no further reduction of nitrate concentration in the anoxic phase beyond 25 minutes, we can reduce the anoxic time. We can also increase the aerobic time as nitrification does not seem to have completed. This figure compares an SBR mimicking MLE system and an actual MLE system, which is represented by a dashed line. The effect of SRT is very similar. Because SBR behaves more like PFR, they have slightly sharper transitions of performance with the increase of SRT compared to the actual MLE system in each zone. The plug flow-like performance for SBR results in a more complete ammonia removal and slightly higher allotrophic biomass as shown in panels B and F. This graph shows the effect of recirculation ratio on the reactor performance. The trend of reactor performance over different recirculation ratio for SBR is very similar to that of the MLE system. The aerobic fraction of SBR is the time fraction for aeration, but has the same effect on SRT. And again, it is very similar to the actual MLE system that it mimics. This last nitrogen removal system I want to briefly talk about is the separate stage denitrification. It is a standalone anoxic aerobic system with no recirculating mixed liquor, used exclusively for denitrification after a separate nitrification process. This is typically used to upgrade the conventional plants that uses aerobic processes that already have long enough SRT and high enough oxygen supply to achieve nitrification, or some industrial wastewater that contains little ammonia but a lot of nitrate for nitrogen removal capabilities. Because it receives aerobic effluent that contains little organic substrates, organic carbon is usually dosed at the beginning to provide electron donor for the denitrifiers. A final aerobic stage is used to scrub organic carbon and enhance sludge settlability. The major drawback is that the supplementation of carbon source can be expensive. It is a compromise because it can be much more expensive to upgrade an existing plant by tearing down the old reactor and constructing a new one. But there has been some study trying to divert the acetate produced during the acid phase of an anaerobic digester as the carbon source. For phosphorus removal, the simplest configuration is the AO process that has an anaerobic zone followed by an aerobic zone. The sludge containing PaO is recycled so that they are in an alternating anaerobic-aerobic environment, providing a competitive advantage for PaOs and resulting in a low effluent phosphorus concentration. But if nitrification occurs in the aerobic zone and nitrate gets into the anaerobic zone through the recycled flow of biomass, less acetate will be produced from the fermentation process, and PaO no longer have the environmental advantage to outcompete the ordinary heterotrophs. A relatively short SRT of 2 to 4 days is maintained to minimize nitrification. Let's look at the effect of SRT on the performance of AO system and compare it with a single aerobic CSTR, which is represented by the dashed line. As a result of the anaerobic-aerobic configuration, a large proportion of biomass in the AO system is PaOs, as shown in panels D and F. They effectively removed phosphate, as shown in panel A. Moreover, if nitrate is produced in aerobic zone at high SRT, the denitrifying PaOs can remove the nitrate that is recycled in the anaerobic zone, along with the biomass. Like MOE system, because the aerobic zone is a fraction of the overall volume, a longer SRT is needed in the AO system to achieve nitrification compared to a single aerobic CSTR. As I mentioned before, the recycled biomass also carries nitrate produced through nitrification in the aerobic zone back to the anaerobic zone. The higher the biomass recycle rate, more nitrate will be recycled, causing PaO be outcompeted by ordinary heterotrophs and less phosphate removal. As for the volume ratio of the anaerobic zone and aerobic zone, the volume ratio determines SRT ratio, similar to what we have seen in the MLE system. If the aerobic fraction is too large, nitrification can occur due to the long SRT, reducing the phosphate removal efficiency. If the anaerobic fraction is too large, fermentation may continue after PaO have taken enough acetate that they need, leaving soluble substrate to the aerobic zone. This can mitigate the competitive advantage of PaOs over ordinary heterotrophs, which is shown in this figure. It is possible to achieve both nitrogen and phosphorus removal within one system, but it is often very complex as it involves anaerobic, anoxic, and aerobic processes, and there are antagonistic effects between nitrification and phosphorus removal. Therefore, it is difficult to achieve good nitrogen and phosphorus removal at the same time. A2O system, as an example, is essentially the combination of MLE process and AO process. The effluent first gets into an anaerobic zone for acetate uptake by PAOs, followed by an anoxic zone for denitrification with recirculated nitrate as the electron acceptor. Finally, an aerobic zone for phosphorus uptake and nitrification. A2O process has the same drawback of incomplete nitrogen removal as the MLE system because a portion of the nitrate is discharged from the aerobic zone without denitrification. Additionally, nitrate is also present in the recirculation biomass flow to the anaerobic zone, reducing the VFA production from fermentation, therefore the phosphorus removal efficiency, especially when there is not sufficient soluble organics in the effluent. 5-stage bottom flow is essentially adding an anaerobic zone at the beginning of a 4-stage bottom flow for phosphorus removal. Like 4-stage bottom flow, 5-stage bottom flow system also achieve high nitrogen removal rate through more complete denitrification in the last 2 stages. But its phosphorus removal efficiency is not improved upon the A2O system. On contrary, sometimes even poorer phosphorus removal is seen from the 5-stage bottom flow systems as a result of a low sludge production and wastage rate due to the long SRT. Remember that the PAOs accumulate phosphate in the biomass. Phosphate is truly removed when the biomass is removed. The large and complex reactor can also be costly to construct. University of Cape Town process and Virginia Initiative plant have essentially the same configuration. VIP operates more like a PFR at a high rate and shorter SRT. There are totally 3 lines of recirculation. The nitrified recirculation, NR, recycles the nitrate-rich mixed liquor back to the anoxic zone for denitrification, while the recycled activated sludge also gets returned to the anoxic zone. So the nitrate that can decrease the efficiency of phosphorus removal is denitrified in the anoxic zone before being returned to the anaerobic zone through the anoxic recirculation AR. The recirculation design ensures high phosphorus removal efficiency, but like MLE, the unrecirculated nitrate is discharged. Therefore, it generally cannot achieve high level of nitrogen removal. Also, the addition of recirculation line can consume a lot of energy and can decrease the reaction rate by diluting the substrate. The Johannesburg process has a similar concept as the UCT or VIP processes. The nitrate in the recirculation flow is first removed before entering the anaerobic zone, ensuring a high phosphorus removal rate. It achieves this by adding an additional anoxic zone before the anaerobic zone. The effluent that contains organics, however, is fed directly to the anaerobic zone to support the growth of the PAOs. This is another process that focuses on phosphorus removal and allows some nitrate present in the effluent. The additional anoxic zone replaced one recirculation line, so less energy is needed for recirculation, but more construction cost is needed. These are just some examples of suspended growth biological nutrient removal systems. There are far more novel designs that we have introduced here. Knowing the principle, you should be able to understand their advantages and limitations by analyzing their design configurations and operating strategies. One last note. If both low effluent nitrogen and phosphorus concentration need to be achieved, you can focus on biological nitrogen removal and precipitate the residual phosphate chemically by adding ferric ion.